Welcome back to today's lecture, Erythropoiesis. I'm your presenter, Dr. Mausmi Shankaradala, working as an assistant professor in the Department of Biochemistry, St. Joseph's College for Women Autonomous. Today's lecture is a part of the certificate course named General Physiology, conducted by Department of Biochemistry, St. Joseph's College for Women Autonomous, Vishakhapatnam. The course is a duration of 8 weeks. Uh, the candidates may be degree, first year to final year students. Contents include introduction of the topic, the site of erythropoiesis, origin of the cells, progenitor cells, stages of the development, hemoglobin formation, loss of nucleus, clinical importance, and in the end we'll be concluding with a summary. Erythropoiesis. What exactly is erythropoiesis? It is just a basic definition of formation of red blood cells. I repeat, dear students, erythropoiesis is the process of formation of red blood cells. It ensures a continuous supply of erythrocytes in circulation. It is essential for maintaining oxygen transport in the body. The process occurs throughout the life in human because it helps to maintain the normal physiological balance. It also compensates for the destruction of old RBCs. As we all know, red blood cells, the lifespan is 120 days. So once a red blood cell takes birth, exactly on 120th day, it is going to be seized off, like removed from the blood. So definitely every single day from the birth to death, this erythropoiesis has to continue. It's mandatory default. It is a highly regulated biological process and it is vital for survival and cellular respiration. So why are we saying vital? Because blood is the only component which carries oxygen. It is the only component which carries carbon dioxide. So unless until you have this carbon dioxide and oxygen, we cannot survive. So in order to manage this, the blood cells are very important. So that is the reason we are saying right from your throughout your life, right from the birth to death, the erythropoiesis has to continue. And we have already seen here it occurs in the red bone marrow. Definition. Coming to the sites of erythropoiesis, in adults it occurs in red bone marrow. Major bones which include like sternum, ribs and pelvis. There the red bone marrow, this it is a major site of this erythropoiesis. Whereas in embryos, it occurs in the yolk sac where the zygote is formed, then the embryo. In fetus, okay, when the baby develops, like the days baby inside the womb, like weeks, we can say. In fetus, liver and spleen are the major sites. Again, once again, I repeat, in adults, the bone marrow is a site for erythropoiesis. In embryos, the yolk sac, whereas in fetus, the liver and spleen are the major sites. With the passage of the age, like as we age older, the sites also will be changed. Bone marrow becomes only the primary site after birth. So when once the baby takes the birth, no liver, no spleen, no yolk sac in that animals and all. So here we see that only bone marrow will be the primary site for the birth, after the birth, for the erythropoiesis. It is restricted to specific bones in adults. Why? Because to ensure efficient RBC production. Origin of cells, the process starts from hematopoietic stem cells. These cells are multipotent in nature. I repeat, these are multipotent in nature. So they differentiate into erythroid lineage. So the hematopoietic stem cells which are present, they can divide, okay, multipotent. So they can differentiate into number of erythroid lineage. Now these stem cells reside in the bone marrow, right? You are having bone marrow. Inside them the stem cells are present. They undergo controlled division, not uncontrolled, controlled division. This gives rise to progenitor cells. Again, I repeat, dear students, we have the bone marrow. Inside the bone marrow, the stem cells are present. These stem cells undergo controlled division. Okay, these cells which are formed after the division are called as progenitor cells. This is the first step in RBC formation. This ensures the continuity of blood cell production. Now, what happens to these progenitor cells? Erythroid progenitor cells developed from the stem cells, as we already seen in the previous slide. They are committed for the RBC formation. Now, these progenitor cells, which one the stem cells divide into progenitor cells, they undergo rapid cell division. They differentiate into proerythroblasts. Now, the progenitor cells from the stem cells are converted into proerythroblasts. These are sensitive to erythropoietin. Okay, the proerythroblasts are very sensitive to a protein called erythropoietin. They increase in the number during hypoxia. Hypoxia means less level of oxygen. So inside your bone marrow, stem cells are present. They divide to form your progenitor cells. These progenitors further divide to form proerythroblasts. Usually these proerythroblasts are very sensitive to erythropoietin, but they increase during hypoxia. They form intermediate stages of RBC. Who? Proerythroblasts. They form intermediate stages of RBC. These all are essential for erythropoiesis. Already we read in the first definition. What is erythropoiesis? The formation of RBC. Now, the proerythroblast is the earliest RBC precursor. It has a very large nucleus. Can you see this point here? What we read right from our school days, RBC has no nucleus at all. But here you can see the precursor means from whom the RBC is going to be formed. It is having a very large nucleus. Cytoplasm is basophilic in nature. Okay, it has a cytoplasm. Obviously, it is a cell. The cytoplasm is basophilic in nature. It actively divides to form more cells. Now, it begins hemoglobin synthesis. So proerythroblast is having a nucleus, then hemoglobin synthesis occurs. Why? The RBC should contain only hemoglobin, nothing else will be present. It marks the beginning of differentiation. Now further stages are formed. Now all these are occurring inside the bone marrow. Stages of development. What are the stages of development? The cells pass through multiple stages. Okay. The basophilic erythroblast stage follows. First one, basophilic erythroblast. After that, polychromatophilic stage. Usually this one shows mixed staining actually. Okay. First one is basophilic erythroblast. Proerythroblast is converted to basophilic erythroblast. From here, polychromatic chromatophilic stage, then orthochromatic stage. It has more hemoglobin. The nucleus becomes condensed. Okay, the nucleus becomes condensed. Gradually, changes occurs in cytoplasm. Now what happens? The cells become smaller in size. Each stage leads to maturation. On the other hand, hemoglobin synthesis increases progressively. So the iron is essential part of hemoglobin formation. We all know hemoglobin is a combination of heme and globin. Globin is a protein, whereas heme is a porphyrin which is having iron in the what you call center place. So iron is essential for the hemoglobin formation. It gives red color to the red blood cells. Now this red color occurs to the red blood cells because of the hemoglobin, and it also enables the oxygen transport. Already we know hemoglobin combines with oxygen to form oxyhemoglobin. Then it will carry that to the lungs. There it will collect the carbon dioxide. It becomes carboxyhemoglobin and again comes back. Now this synthesis begins everything in early stages. It continues till maturity. Now there is a very well-known deficiency called as anemia. Anemia means blood. Anemia means having less blood. This deficiency occurs when there is no ample amount of blood in your body. It is crucial for RBC functioning. Loss of nucleus. Nucleus is lost during maturation. Okay. Where in RBC initially nucleus is present. Later on it is lost. Now this increases space for the hemoglobin. Thus, it improves oxygen carrying capacity. The main characteristic of mammalian red blood cells is this only, like it has space and it has hemoglobin carrying. The cells become more flexible, okay, than the regular cells. It helps in capillary passage, definitely, because the blood vessels are very thin, they're very narrow. So all this, if the nucleus is lost, then the cells becomes more flexible for going into the capillary passage. It marks near final stage of development, and it is an important adaptation, loss of nucleus. Reticulocyte stage. That reticulocyte stage is immature RBC. It lacks nucleus but has organelles. It enters the blood flow from the bone marrow. That is, in the bone marrow till now we have come up to this reticulocyte. From here it enters the bloodstream. Okay. It matures within 1 to 2 days. It completes the development in the circulation itself. The immature last stage, that is reticulocyte, is a immature RBC. Already the nucleus is lost and hemoglobin is already entered into that, and it enters the blood flow. In 1 or 2 days, inside the blood flow only, bloodstream only, they will become mature. It completes the development in circulation. Got it? It still synthesizes small amounts of protein. This transforms into mature RBC. So this is the transitional stage. So from the reticulocyte, it enters the blood flow. There it will be transferred into the RBC. How a mature RBC is fully formed? It lacks nucleus and organelles. It contains only abundant hemoglobin, and it is already, we know, biconcave in shape. It transfers very efficiently oxygen, and it circulates for about 120 days. It is flexible and durable, and it performs very vital functions. Regulation, erythropoiesis is hormonally regulated. Erythropoietin is the main hormone. It controls the RBC production rate, okay, whether it is required, whether the red blood cells rate is, the required or not, it will be regulating. It responds to the oxygen level. Already we have read initial stages. When the oxygen is not present, the condition is called hypoxia. During this procedure, what happens? More amount of that production occurs, okay. So it responds to oxygen levels. Low oxygen increases the production, vice versa, high oxygen reduces the production. It maintains homeostasis and it ensures proper oxygen supply. Erythropoietin, this is the hormone. It is mainly produced by the kidneys. Again, I repeat, erythropoietin hormone is produced by the kidneys and it is mainly released during hypoxia. Erythropoietin, what it does, it is making the erythropoiesis occurring. Erythropoietin is the hormone. It helps in the procedure called erythropoiesis. Erythropoiesis means formation of RBC. So when there is no oxygen, definitely more amount of RBC has to be produced. Who will produce that? This hormone will give the indication. So that is the reason we understood during hypoxia, when no oxygen is present, this hormone is released by the kidneys. Therefore, the RBC production will be increased. So this acts as progenitor cells. And this is essential for normal erythropoiesis as well as any abnormal conditions. Deficiency reduces the RBC count and it is vital for regulation. Nutritional factors. As we already studied, iron is very important for this hemoglobin. Along with this, vitamin B12, that is cyanocobalamin, is essential for cell division. Why? Because here we are seeing continuous cell divisions. Folic acid aids in DNA synthesis. So this is the reason the pregnant women are usually prescribed to take this folic acid. Even in government initiative also, must and should every pregnant woman, irrespective of their economical background, they are supposed to take the folic acid. Why? Because it undergoes this DNA synthesis where the cell division and all occurs. Deficiency affects RBC formation and it leads to a condition called anemia. N means not there, anemia means blood. Balanced diet is very, very, very essential. Nutrient supports proper development. So they ensure healthy erythropoiesis. Clinical importance, erythropoiesis maintains oxygen balance, defects lead to anemia, excess leads to polycythemia, okay, excess production. It is important in medical diagnosis and used to study blood disorders. It also helps to understand physiological changes, important in treatment planning and essential for overall health. Summary, same, the erythropoiesis maintains oxygen balance and defects leads to anemia. It helps in understanding physiological changes, important for treatment planning, essential for overall health. Summing up the references, Guyton and Hall textbook, we have both national and international editions here, like textbook of medical biochemistry and physiology by Chatterjee and Shinde, and human anatomy by Allen and Markey, and essentials of medical physiology by Shimbalinga. Thank you.